Good morning, students. The topic for today is phenocopy, pleiotropy, and multiple alleles. To begin with, what is phenocopy? When the changes in phenotype are altered by environment, then the new phenotype resembles another phenotype produced by known genes, which is not inherited, is called as phenocopy. It may be affected by temperature, radiations, chemicals, etc. The term phenocopy was coined by Richard Goldsmith in 1935. The example for the phenocopy is the Himalayan rabbit, which develops black coat color if it lives in cold environment. It is a phenocopy of genetically black rabbits. If both are made to live together at high temperature, then the Himalayan rabbit would have a different phenotype than the genetically black rabbit. Another example of phenocopy is the Thalidomide babies born in West Germany and Great Britain in 1960s. Deformed children were born to mothers who had taken thalidomide drug during their pregnancy. Similar deformities were seen by another phenotype, phocomelia, caused by a recessive gene. Moving on to pleiotropy. Although a change in single enzyme usually disrupts a single biochemical pathway, it frequently has more than one effect on phenotype. So, we can say that when one gene influences multiple phenotypic traits, it is called as pleiotropy. Mutation in a pleiotropic gene may have an effect on some or all the traits. Genes which show pleiotropy produce a single polypeptide just like other non-pleiotropic genes, but it governs such a biochemical reaction which is basic to many developmental events. The term pleiotropy was coined by Fetchcraft in 1910. It is a Greek word in which pleio means more and tropy means character. The classical example of the pleiotropy is the human disease phenylketonuria, which causes mental retardation and reduced hair and skin pigmentation. Phenylketonuria is due to the mutation in a single gene that codes for the enzyme phenylalanine hydroxylase. This enzyme converts amino acid to tyrosine. Due to mutation, conversion of phenylalanine to tyrosine is reduced or ceases completely. Phenylalanine in bloodstream is toxic and causes mental retardation, whereas tyrosine is used by the body to make melanin. The failure to convert normal levels of phenylalanine to tyrosine results in less pigmentation of hair and skin. Moving on to multiple alleles. Multiple alleles. When more than two different forms of a given gene exist in a species, they are called as multiple alleles. Normally, according to Mendel, we say that there are only two alleles of a particular trait. There can be large number of possible allelic forms in that same population. This situation is called as multiple allelism. The multiple alleles, they always occupy the same locus on the chromosome and influence the same character, and no crossing over occurs among the members of alleles of the same multiple allelic series. Examples of multiple alleles. The most common example is the blood group ABO in human, coat color in mice, coat color in rabbit, eye color in Drosophila. The classical example and the most common example of multiple alleles is ABO blood group of human beings. Gene coding blood group has 3 alleles rather than 2. For ABO gene, the 3 alleles are IA, IB and I alleles. We write these alleles as A, B and O alleles, where I stands for immunoglobulin. IA and IB show codominance, which means an individual who is heterozygous for these two alleles, the phenotypes of both alleles are completely expressed, thus producing AB blood type. The I allele or the O allele is recessive to both IA and IB alleles. Number of genotypes in a population depends on the number of alleles. So we can calculate the number of genotypes with the help of n plus 1 divided by 2, where n equals the number of different alleles at a locus. Next example of multiple alleles is the coat color in mice, which is determined by a single gene with a series of alleles.